want the whole uh, that is cross. Who is the parent? What are the gametes? What are the generations? And what is there in the F1 or F2 generations? Not just the chart. Okay. I need the whole information. That is the first thing. Second thing is that when you are determining that Sally has this kind of genotype or Sally's mother has this kind of genotype, you need to explain that. Why? Why? That is the main reason what we are doing here. I know that if we do this kind of cross, it will happen. Not the Punnett square is not the whole cross. Okay. I need the whole cross from parent to end. Time bound for this one. Just complete this and send it back. Okay. Even for some kind of homework 3 also, I didn't find the whole cross. I need the parents, the generation, gametes, which gamete is from which parent, that is male or female, and everything. Another thing is that do not mix the pedigree with this one. In pedigree, the symbol is this, but that is only for pedigree. Okay? Do not mix this one with the cross. In cross, it is this. Okay? Either or write MF. That is also acceptable. You can write F or M, whatever it is. But this is exclusively for pedigree. Okay. So these are a couple of things, and right now I need one lucky person from each group.
I'll do it. I'll
One
do
lucky
it for
person
us.
from each group. Now you decide who is the luckiest in your group.
I'll volunteer for you.
And come here. One person from each group. And take this one, based on this one.
I gotta do it for my group.
Only one.
Only one piece of paper.
Tell me the number. I'm doing double cross.
2.
2. Who is 1? 4.
1.
1. Take it. Take one from here. Now 2. 3. And 4. Okay. Let's see. That's great. That's great. What is yours? Okay. That's great. So you have assembly. Okay. That's fine. You three are fine. I think so. Do you want to do SMP or anyone HPV or HIV?
I'll take it.
I think you do HIV because HIV is in your course. So just do HIV. That is fine with you. Okay. So one thing is that for this this project, I have assigned the project for your group. The first and foremost thing is that go to Wikipedia. Okay, but do not do anything from Wikipedia. Just go read it thoroughly. In the Wikipedia, you will find some kind of that is references there. Just under the Wikipedia. Okay. Go there. Wikipedia is the best thing to have some good idea about the whole thing, the basic idea about the whole thing. But it is not at all. That is, there are so many other things there. Go to the references there. From the references, you will find different kind of things. Okay. Suppose for P53. P53 is the most that is important molecule in modern biology, in modern science, P53. And it has definite various roles for cancer formation, retinoblastoma, breast cancer, prostate cancer, and everywhere. That is the cell signaling. It is controlling the whole process. Okay. So after getting the knowledge about that one, just the basic knowledge about that one, what is the molecule, what it is doing about, after that, go to some papers. Okay. Then you will find the whole thing. Main part is there for the project. We don't know anything. All the students. Okay. These subjects are hot topics in your that is in modern biology. Okay. Another one was there, but it will be too hot and too advanced for that one. That is PDL. PDL is the right now it is the hot topic in cancer research. PDL1 and PDL1 inhibitor. Okay. So like that, have some idea about that. Then go to one website. I don't know whether I told you or not. That is called PubMed. PubMed. Another is GeneMed, and go to NIH website. PubMed is actually NIH website. Okay. There you will find a lot of advanced and current research. Even in PubMed, within a couple of minutes, you will find 10 new papers there. Those are the real scientific papers. Okay. But that is after you get the idea about the whole thing. In book, you will find a little bit of information about that. For HIV one, the main thing is that HIV is the retrovirus. Okay, this retrovirus actually incorporates its DNA into host DNA, that is the human DNA, and it is creating that is damaging the lymphocytes. Okay, that procedure we need to know that procedure very well. Okay, for HPV, this is also a kind of virus, and it is creating the cervical cancer. For HPV groups, and it is very important to go for some kind of epidemiological work because this Claremont County is within Appalachian region. In the whole USA, this Appalachian region has the highest incidence of HPV, human papilloma virus, and cervical cancer. I already did some work on that. Okay, and another group is there in the Kentucky, and she is actually devoted her whole research into this HPV one. So you'll get a lot of information from there, and just that is go with me. That pages are there. I will update the groups because I need to include these four in one group. There, collect the informations. Our main target is that to find out the mechanism of that. Okay, how the HPV is working, how P53 is working, how that HIV is working. Okay, HIV, HPV is in your syllabus, but I will not cover the bacterial part a little more. P53 is everywhere, everywhere, and you have telomere. That is another very important thing. Actually, I had another project on gene therapy, and telomere is one most important thing is that telomere actually protects the chromosome. Telomere is the end part of the chromosome, two end parts. Okay, it is protecting the chromosome from damage, and telomere has repetitive sequences. Telomere transcription, telomere replication is different than the normal DNA. Okay, the main thing is that during replication, telomere gets shortened each and every time. It is losing one or two nucleotides or one or two sequences, something like that. So that is the thing is that when we are getting aged, that time the telomere size of telomere is very short, and that is prone to other infection. Not only that, even that can create cancer. So the telomere means aging and cancer. Okay, so these are the things. I think these are very interesting topics. When you will go for the research, you will find a lot of information, and these are the most hot topics in modern biology. Okay, and I am always with you. So in your group project there, in your group page, you have file. So it is your presentation. So if you want to do the whole thing in PowerPoint, starting from beginning from PowerPoint, you can do that. But it is better just go find out the points. In what point you need to discuss the whole thing. Have some information first, then jot down the points that these are the points I need to talk about. Then gather information and make the PowerPoints. Okay. Main thing is the mechanism. How the retrovirus is working, how the lymphocyte is getting damaged. These kind of things we need to know. These are the all molecular biology part. and a little bit of epidemiology, that is the where is the most prevalent cases are there, and a little bit of treatment and other things. Because particularly for HIV, we have a lot of new informations, that is the vaccination and other things. HPV, we have the vaccines. So how the vaccine works and at what age the vaccine is needed, these are the informations, little bit, just one or two slides about that. Main thing is the mechanism. Genetics part, mainly. Genetics and molecular biology. Okay. Okay, these are the things. And today we are going to continue that part, that is the chromosomal aberration. But the thing is that this is a little bit long chapter. This is a little bit long chapter, and I am modifying the slides a little bit. So I put the slide and put one there. Okay. This is not the final slide. When I will finish the slide, I will upload the finished slide with including these slides also. So that will be the final. So for today, you can use that one, but later I will modify it a little bit more. So we already talked about this one, karyotyping, banding, and Q band, G band, C band, R bands, everything. We talk a little bit about the deletion also, but I will cover the deletion part today also a little bit. Okay. So we have generally two types of chromosomal aberrations. Chromosomal aberrations means any kind of change in chromosome, either in structure or in number. So if the change is within the structure, then it is called the structural aberration or rearrangement, chromosomal rearrangement. Okay. And due to this one, the main thing is that there should be some kind of damage and reunion there. Okay. And numerical aberrations means change in number. Change in number means either one chromosome is extra or the whole set is extra. So like we have 23 pairs, 46 chromosome, 46 plus 23. That could happen. Or suppose chromosome number 18 is in 3 doses. That is also possible. So these type of aberrations are called numerical aberrations. Okay. So in the structural aberration, we have these 4 kind of things: duplication, deletion, inversion, and translocation. In the translocation part, we have 2 things. One is reciprocal translocation, actually 3 things, and one is Robertsonian translocation. Okay. So the first thing is that what is duplication? Duplication means, suppose here you can find there are 2 genes E and F here. Okay. This E and F, that is the normal chromosome. In this chromosome, we have 2 E and Fs. So E gene and F gene, it has been duplicated here. It could possible. So if this kind of thing is happening there, it is called duplication. Okay. Here E and F gene was there, but in this part, only after D there is G gene. There is no E and F. That part was broken from there, broken from the chromosome, and lost that part. So this is called deletion. Deletion of a part of chromosome. Okay. Now, in this case, DEF, there are 3 genes, but you could find here that DEF in opposite direction. Okay. So that is called inversion. And in this case, we know there is exchange of chromosomal segments between sister chromatids, that is sister chromatid exchange, between two homologous chromosomes, but in non-sister chromatids that is called a crossing over. But here in this case, there are two non-homologous chromosomes. That is, suppose it is chromosome number 9, it is chromosome number 11. But one part from here is moving to that chromosome, some other non-homologous chromosome. This type of transfer is called translocation. So based on the chromosomal segment exchange, there are three types: sister chromatid exchange, crossing over, and translocation. Okay. So all these things are changes in chromosome structure. That's why these are called structural aberration or chromosomal rearrangement. There are two names here, either structural aberration or chromosomal rearrangement. So rearrangement starts with DNA breakage. If there is no such DNA breakage, there is no way they can rearrange, or in one chromosome there is just one strand of DNA is double-stranded. Now think about only DNA. In one chromosome, this is one chromatid. In one chromatid, DNA is coiled up with histone, and DNA is double-stranded. Okay? So if this part breaks, that means the double-strand breakage will occur in the DNA. Clear this part? So there should be double-strand break anyway. And generally, if there is double-strand break, they are potentially lethal if it is not repaired. But we have the repair mechanism in our body, in our cell system. They can repair. If there is some breakage or something, they can repair the whole thing. We have the molecular things there. They will repair. But if it is not repaired, then it is lethal. If the gene is lost, that means it is lethal. So now, in case if the two ends of the same break are rejoined again, so there is a break and it is rejoined again in the same manner, then there is no problem. There was breakage, even we cannot understand that whether there was a break or not. Okay. But if that part goes to somewhere, if that part goes to same chromosome, if that part goes to some different chromosomes, if that part inverted there, then that is structural aberration. So all the concept here in the structural aberration, there is that there should be break first, and that break should be rejoined. During this rejoining, either it goes to some other part of the chromosome, some other chromosome, or it is inverted, then the structural aberration would happen. Okay, clear this part? So it always starts with a break. And another part is that rearrangements can also arise through errors in crossing over. How? Sometimes during crossing over, if the homologous chromosomes are just not by the side by side and the crossing over occurs, then this kind of thing can happen. So what is duplication? Duplication means the process of exchange of chromosomal segment into other chromosome when there are two copies of that chromosome. Two copies of that gene in the same chromosome. That is called the duplication. Here we have the EF gene, but here EF in two doses. So we can say that there is a duplication of EF gene here. Clear? Now the thing is that when the EF gene is there, how can they pair during meiosis? During meiosis, homologous chromosomes comes together. They are homologous, means all the genes matched with each other. C, D, E, F. EF will also match, but what is for this other EF? They can't match because there is no opening there. But the G genes should match with the G1. In such case, they will form this kind of the homologous part. So they matched with each other, all the genes. This part is also matched with each other, but this is extra. There is no extra EF here. So they cannot match. During the prophase of meiosis, prophase 1, when the homologous chromosomes come together. So at that point, if you find this kind of loop in a cell, that means there is a chance of duplication there. because we cannot read the genes like that ABCD in the chromosomes or in the slides. Okay, we can find these kind of loops. So during the pairing, they can form these kind of loops. Now, based on the duplication sites, we have three kinds of duplication. One is called the tandem duplication. Tandem duplication means in the same part, this is the normal chromosome, this is the duplicated part, that is 2 and 3. They are adjacent to the original chromosome. That is the original region. Here are the genes, 2, 3, original region was there. This is the duplicated part, but they are just by the side of the original part. In such case, it is called tandem duplication. If they are on the same chromosome, but in the different part, that could happen. Then it is called non-tandem duplication. Even it could happen that not in this chromosome, in the other homologous chromosome also. In such case, we should call it as non-tandem duplication. This one is called reverse duplication or reverse tandem duplication. Why reverse tandem? Because the duplicated part is here just by the original one. So that is the tandem duplication, but it is reversed. Other than being 2, 3, it is 3, 2. So this type of duplication is called reverse tandem duplication. Now, what is the effect of this duplication? Duplication means double dose of the gene. If it is double dose, then there is the problem with the dosage compensation. What we saw during the bar body formation, 1X is inactive, or those kind of things. That dosage compensation, that kind of problem is here. In case of fruit fly, we have seen this one. One is called the bar mutation. Bar mutation is that it is incomplete mutation, and with this bar gene, this kind of bar eyes are formed. This is the bar eye. What is happening that in the wild type female, the bar region is normal, there is no problem, so it is normal eye. Dosage compensation. Okay, one is in high dose, another in is low dose. In such case, we can find this kind of little bit smaller eye, bar set. Rather than round set, it is bar set. That's why it is called the bar mutation. In case of homozygous condition, when there are double doses, but that is incomplete mutation, in such case, we will also find the bar, but it is a little bit smaller than that one. In case of triple duplication, we could find the bar is very, very small. Okay, so generally, in case of duplication, we will find the problem with dosage. And due to that kind of thing, we can have this kind of phenotypes. If it is within coding region. Now the question is that whether it is within coding region or not. Because in human chromosome, particularly for the telomeric region, there are a lot of genes repeated sequences. Repeat. Suppose 1000 base pair of the DNA segment, it is repeating almost 100 times. So there is a duplication there, but that is non-coding region. There is no protein is formed from that part. So we cannot see any kind of phenotype. But if it is in the coding region, then we could find this kind of things. Now, we told that the unequal crossing over can lead to the development of deletion or duplication. In this case, there are two genes. One is the red gene, one is the green gene. So when they should be aligned, the homologous chromosomes, the red genes should be in the red position, green genes would be aligned. That cross is the crossing over part. If that is the crossing over part, so we can find one red, green, and green in one chromosome after the change of chromosomal segment, and only one red in the other chromosome. Okay, just think about that. Red, green and green. Okay, like this. So in one chromosome, we'll get two green genes, and in one chromosome, there is only one red gene. So there is a duplication of this gene due to unequal crossing over. Okay. And for that one, there are developmental disabilities for this dosage. What is that? In normal condition, there are three proteins formed here. One is red, one is orange, one is yellow. Okay. But if there is duplication here in the B gene, the orange gene is more. They will create more proteins. So it is in double dose. The red gene is in one dose and the yellow gene is in one dose. In such case, if that dose is too much for the orange gene, then they can have some kind of abnormalities here. This is the effect of duplication. Okay. And particularly, they can have severe consequences when the precise balance of gene products in the critical to the cell function. And most of the cases, it is lethal. Most of the cases, it is lethal. So this is in a short about duplication. What is deletion? Now think about that unequal crossing over. So there were two genes, red and green genes, initially on the two chromosomes. After the crossing over, one chromosome got two green genes and one red gene, and another chromosome got only one red gene. Right? That means that is duplicated. One chromosome is duplicated, but another chromosome, that green gene is deleted from that. So that is called deletion. That is one way it can be deleted. Or there could be a break here in this CD gene. There could be a break here, and this part is lost. Okay. In such case, we could have this kind of deletion. Deletion means absence of one chromosomal segment or a certain part of the chromosome. Centromere, this part, if it is other than centrometric part, if it is within this part, there is no problem of cell division. Okay, it can go to the gamete. But if within this part, then it will not go to the gamete. Why?
There's no centromere.
There is no centromere. But why? What is the problem for that? If there is no centromere?
There's nothing for the spindle to attach to.
Okay, so the centromere contains kinetochore, and kinetochore is the position where the spindles are attached. If there is no centromere in this part, this CD part, then the spindle will not attach to that part. So that part is just in the cytoplasm. It will not go anywhere. In such case, we have the cellular mechanism. One organelle will digest these things. In such case, which is excess, which is non-functional. Do you know what is this? An animal cell. One organelle can digest this non-functional part of the cell.
The eosinophil or
Eosinophil is a cell, particular cell in the blood. Eosinophil, basophil, and they have some phagocytic procedure, macrophages, and all those things. But those are the lymphocytic and body defense mechanism. But I am talking about only one cell. In cell, there is one cellular organelle that is doing this kind of things. That is called the suicide bag of cell. If the whole cell is damaged, it can destroy the whole cell. Suicide bag of the cell. Powerhouse of the cell. What is that? Mitochondria. Suicide bag of the cell. Animal cell. It is not in plant cell. Having highly acidic broken part. It can go with the cell, with the mitochondria and other things. But the function is that lysosome will destroy these things. Okay, so it will not go to the gametes or in the that is next generation, F1 generation. So what is the effect? The effect, there are a couple of effects here. One is the imbalance in gene product, definitely. If one gene is in half and one gene is doubled in other chromosome, then there should be some kind of imbalance. Is deleted from that chromosome, that recessive genes which should not occur or should not express, that will occur. And haplosufficiency. Haplosufficiency means only half dose is working. I'll talk about this dominance. And before that, we need to think about what kind of deletion it is. Is it intragenic deletion or it is multigenic deletion? Intragenic means a small part within a gene. It is deleted. A small part of the DNA within a gene. So what is a gene? A gene may be of couple of thousand base pairs. So DNA is measured with the base pairs. Base pairs means DNA is CG combination in two strands. So one of these, A always binds with T, C always binds with G. So one of these together is called a base pair. Okay. So a gene may contain thousands of base pair long DNA. Okay. So suppose in this case only this part got deleted. What will happen? Or only these two nucleotides part got deleted. What will happen? That is also called a kind of point mutation. What will happen in such case? What is the function of gene? So we will talk about this one a little bit later. Probably from the next class. We'll go for this one. What is the function of gene? You know how gene works? Gene synthesizes proteins. Okay. Every three nucleotide together, actually it will form one mRNA, and every three nucleotide together can form one amino acid. So as a whole, the whole gene is creating one polypeptide chain. One amino acid, two amino acids are connected with a peptide bond that is in the protein structure, primary structure of the protein, and they will create the polypeptide chain. If this part is deleted, suppose, then the chance is that what amino acid it could code for this or in original condition, it will code a different kind of amino acid. Or it will not code that amino acid as well. There is no way. So the polypeptide chain will be different. And that is, so inactivate the gene and has the same effect as null mutation of that gene. Null mutation means that is almost lethal. There is no gene there. If the homozygous null phenotype is viable, if it is viable, generally it is not viable, the homozygous deletion also will be viable. And intragenic deletions can be distinguished from mutation caused by single nucleotide changes. Okay. And these kind of mutations never convert to wild type. There is no way that another addition of nucleotide will be there. It could not be the same nucleotide. If that is happening also, it could not be the same nucleotide. Okay. So it cannot go back to the wild type. This is the problem. Actually, the whole gene is damaged in such case. The whole gene is damaged. The functionality is gone for the gene or a different kind of things. And that could create some kind of cancerous genes sometimes. Okay. This kind of mutations. That's fine. What about the multigenic deletion? What is multigenic deletion? Not a single gene, but couple of genes together. It may be the 17, 18 genes together or 100 genes together, the whole part of the chromosome, very long part. All genes are missing from there due to deletion. Generally, in such case, it is lethal. Because if all those genes are missing, they are lethal. And there should be, they have more severe than introgenic deletion, definitely, because couple of genes are missing from there. If such a deletion is made homozygous was inbreeding, the combination is always lethal because the genes are missing. If it is heterozygous for a multigenic deletion, it is having one normal homolog that carries the deletion, that may not survive. So the deletion may uncover deleterious recessive alleles, allowing a single copies to be expressed. Okay, some kind of thing. If there is multigenic deletion, some kind of recessive allele can express. So what is pseudodominance? Pseudodominance means, suppose in this phenotype, this is the gene, that is the chromosome, we have these recessive alleles here. For all recessive alleles, we have the normal wild type alleles here. Okay, in such case, what will be the phenotype? The phenotype will be all plus 1, 2, 3, 4, 5, 6, 7, 7 pluses, 7 normal. That will be the phenotype. This will be the phenotype because recessive gene will not express because dominant genes are there. If there is a deletion between B and C gene, so the B and C gene is not there, the wild type genes are not there. If the wild type genes are not there, only B and C, they don't have any kind of wild type gene to protect their expression, then they will express. So the phenotype will be like this, plus BC and all plus. That will be our phenotype. So in such case, that is a chance that there should be no such expression of BC gene, but we are getting BC gene just due to the deletion of this part. This is called pseudodominance. It should not be dominant, but it is dominating right now. That's why it is called pseudodominance. And that is always for deletion of that part in the chromosome. Again, the formation of loops. When the deleted part is there, just like the duplicated part, when there is deletion, that means in other homolog, there is the extra copy of that part, which is deleted from here. Here's something like T part is here, EF part is missing here. So what will happen during prophase? They will form the loop because G, they need to match with the G here, they need to match with the D here, but EF part is missing from this deleted chromosome. So they will form this kind of loops. And that is visible always. This is called the deletion loop. And this is actually the normal chromosome of salivary gland chromosome of Drosophila. Do you know what is the name of that one? That is the most widest chromosome, longest chromosome. Still we found. Name is there. That is called the polytene chromosome. Polytene chromosome from the salivary gland of Drosophila. These polytene chromosome, they have different kind of bands which denotes actually their genes. Well, from here you could find that this band is matching with this one and this band is matching with this one, just two side by side. And rest of these bands are missing into this chromosome. So it is creating, this is the normal picture from the slide that will create this kind of loop. It is indicating that these genes are missing here. So there is a deletion into this chromosome. So deletion and duplication loops, they can show us whether there is any deleted part or duplicated part in the chromosome or not. We can just seeing that loop, we can identify whether it is deletion or duplication. And what is the effect in the phenotype? This is called notch wing of Drosophila. This is the Drosophila wing, this is the normal wing, normal shape. and this is a notch here. And this notch wing is for notch gene. Okay. And the notch, if there is a deletion of the notch gene, that part, generally that is lethal. If it is haplosufficient, that is, it is in the X chromosome, either homozygous females are lethal, males, if it is on the X chromosomes, males are hemizygous, that is X and Y. So in the half dose there, it is lethal. But if it is heterozygous for the female, that one deletion is there and another not gene is there, in such case, generally we identify this one as this. There is a gap in the chromosome. It is showing the notch character. So if these two X chromosomes are here in the female, one is notch and another that is the deleted part, another the normal gene is there. Then in such case, we will find these phenotypes. They are viable, but the phenotype is different. But generally, deletion, duplication, those are not viable. In case of human, we have a lot of deleted abnormalities. And these are a lot. Deletion and duplication. So couple of these are, one is that cry-do-chat syndrome. Cry-do-chat syndrome, crying like cats. Okay. So in such case, what is happening here? This is chromosome number 5. In the chromosome number 5, in the short arm of the chromosome number 5, there is a deleted part in the cry-do-chat syndrome person. There is a deletion there. That deletion can create this kind of abnormalities. So the people, generally all syndromes, that is genetically deleted or duplicated or chromosomal abnormalities are there, that is numerical aberrations are there, all the patients are generally mentally retarded. Most of the cases, they are not fertile, sterile. These are the common things. Mental retardation is the common thing, and the changes in faces and some other abnormalities are there. So the deletion in the 5p15.2. So we mapped the gene, and that is the 5p means p is the small arm of the chromosome and q is the longest arm of the chromosome from centromere. So it is in the p part and 15.2 position. If there is a deletion in that part, then it can create Cry-do-chat syndrome. Those genes are missing. Another is Williams syndrome. What is happening here? There are two PMS gene here, and PMS actually codes for DNA repair protein. In between two PMS genes, there are 17 genes are there, and those are normal, in normal condition. Okay. If there is unequal crossing over, because there are two sets of PMS here, so in such case, if they can mismatch with one PMS, it should be here. It could bind with that one, but it is binding with this one. That could happen. If there is some kind of mismatch repairing, then there is unequal crossing over. If there is such kind of unequal crossing over, then one chromosome will contain two duplicated part, the 17 gene. Another chromosome don't have that 17 gene part. Okay. This deleted chromosome can, if it is in the gamete and goes to the offspring, then it could create the Williams syndrome. And generally the same thing, mental retardation, certain kind of face patterns and other things. And generally most of the cases, they are sterile. Very few cases they're fertile, and generally their lifespan is within 20, 30 years. In case of Turner syndrome and Klinefelter syndrome, their lifespan is a little bit higher. But Downs and Williams syndrome, Cry-do-chat syndrome, their lifespan is less than that. But right now we have different kind of treatments, so we increase the lifespan a little bit. Okay, so these are all about deletion. Now the inversion. Inversion means the chromosomal segment was there was break, but it got to the chromosome in inverted order. One thing is that this is a very interesting thing. Human chromosome number 5, 4, and chimpanzee chromosome number 4. The difference between these two chromosomes are there. This part in the chimpanzee chromosome is inverted. All other genes are same. All the genes are same. Even not other. All the genes are same because this is only inverted. But that doesn't mean there is a deletion or duplication of the gene. The genes are there. Just the segment is inverted. Okay. So this is called inversion. Just turned 180 degrees. And the inversion means it should have the two breakages there on the chromosome. Then it got just changed. So based on the inversion part in the chromosome, we have two types. One is called paracentric inversion. One is called pericentric inversion. Peri means taking centromere. Para means outside of centromere. So if the inversion is outside of the centromere, then it is called paracentric inversion. If the inversion is within taking the centromere, within the centromeric region, then it is called pericentric inversion. Now, basically, this one, in case of inversion, if there is no alteration of genes are there, just their position is different. Okay. So what is happening? There should not be anything because the genes are creating proteins, all the genes are there, they will create the protein. There should not be any kind of problem. But we have some kind of problem that is called the position effect. What is the position effect is that there is one example in wild type allele of Drosophila, they normally encode the red eyes, these genes. If this gene is going to the white locus, the white locus is recessive, red is dominant there. If this gene, the inverted part, goes to the white locus, what will happen? So the red gene is there also, white gene is there also, and it is creating some kind of different kind of pattern because Drosophila eyes are made up of just like when we are thinking about digital picture, is made up of pixels. Pixels means just spots, okay? And Drosophila eyes are made up of this kind of things. So some of are red, some of are white. That will be a phenomenon, a mosaic pattern. That is called the position effect. So the red gene was there, it inverted and got to the white gene part, it was attached to that part. As a result of that one, both gene expressed and created this kind of mosaic eye pattern. This is called the position effect. Now the thing is that if the gene is inverted or that part of the chromosome, some lot of genes are inverted, what will happen during meiosis? During meiosis, they should pair with the homologous chromosome. Same genes are there, but it is inverted. In case of deletion, we thought one chromosome there was no gene, another chromosome there was the gene was present. So there is a loop. In duplication, in one chromosome there are double doses, in one chromosome there is only single dose. There was a loop. What will happen for this one? And there are a lot of things. Generally, they are forming non-viable springs, that is gametes. This is like that. Okay. So these are two genes, which is one is normal, one is inverted. Okay. So what is happening here? I just need to draw, otherwise. So one gene is like this. This is the normal gene. A, B, C, E, and E. Okay. Another gene is inverted. A, B, after B, so it is the normal condition. If it is inverted region, Okay. Now, if that chromosome will match, so after B, that will be the D part. After B, that is D. So this chromosome will go here. So this is the D part. Then will be C. This is the C part. And then it is E. Centromere is outside of the loop. If it is pericentric, centromere will be here. Okay. So if we will find this kind of loop in metaphase or prophase, during prophase generally, then there is a chance that there is inversion here. Okay, that's fine. What will happen if there is a crossing over here? Suppose there is a crossing over here between these two. Can I figure out that one? So there is a crossing over here, that means A, B. Like this. In such case, what will be the result? So this chromosome or the red one, A, B, C, D, B, and A. What will happen for the other one? So it starts with E, and here it will go to C, D, and E. E, C, D. Basically, there are missing genes here. In one case, we have the duplication AB and AB, reverse tandem duplication almost, or non-tandem duplication, but in reverse position. And in other gene, there is one part is duplicated, the other parts are missing. In such case, if there is no such genes, those gametes are non-viable. Those gametes are non-viable. In such case, but other two are normal. If you think about the other two part of the chromosome, this is normal, and this one is also normal. Okay, those two are normal. One is inverted, one is normal. So we can find out this kind of things. Now the position of centromere. If the centromere is here between A and B, what will happen? So here between A and B, one is one centromere, and here it is one centromere, right? Now, what will happen during anaphase? If there are two centromeres and here there is no centromere at all, this is eccentric fragment. If this is eccentric fragment, it will be lost because it will not attach to the spindle fibers. But what will happen into these two? This chromosome. There are two centromeres there. That will create, if one spindle is here, one centromere is attached here, that is like A, and here is B. Another centromere is attached here, like this. So it will create this kind of breach. This is called dicentric breach. And what will happen? Generally, if those two spindles will pull each other on their sides, it will break from the middle. From where we don't know. Any part of them. Okay. Let's see.
Chromosomal inversion refers to a segment of a chromosome being turned 180 degrees. In this pair of homologous chromosomes, the boxed region of one of the chromosomes will undergo inversion. Notice the position of the centromere and how the inversion of the segment does not include the centromeric region. This is a paracentric inversion. Para means next to. Upon inversion of the segment that contains genes C, D, and E, the chromosome pair becomes heterozygous. Inversion of a chromosome can have various consequences, but specifically, it can result in serious problems during meiosis when an individual is heterozygous for a particular inversion. During prophase 1 of meiosis, the inversion results in the inversion loop configuration shown here, which allows the homologous sequences to align. After forming an inversion loop, a single crossover event can take place within the inverted region during prophase 1 of meiosis. As the chromosomes separate during anaphase 1 of meiosis, you start to see the unusual chromatids that have been formed. One of the chromatids has two centromeres that are connected by a dicentric bridge. Another lacks a centromere. It is an acentric chromatid. As anaphase 1 continues, the centromeres continue to separate, stretching the dicentric bridge and splitting the chromatid apart. The acentric chromatid will be lost in the cytoplasm because the mitotic spindle cannot attach to a chromatid without a proper centromere. Of the resulting four gametes, one will be a normal, non-recombinant gamete, and one will be a non-recombinant gamete with a paracentric inversion. The splitting of the dicentric bridge gives rise to two recombinant gametes that are non-viable because they are missing a significant number of genes.
Clear this part? What will happen if that is, so this is one is eccentric chromosome and one is dicentric chromosome. Dicentric chromosome is having two centromeres. Eccentric, there is no centromere at all. In case of dicentric chromosome, they will form dicentric bridge during anaphase, and there is a chance during cell division that they could break in any part. We don't know in which part, and they will produce two gametes with less reduction of significant number of genes that could result in non-viability of those gametes. What will happen in case of pedicentric inversion? The same thing, it is in pedicentric inversion. So the centromere part is here. In such case, what is happening here? This part is creating one gamete, and this part is creating another gamete. So we will have normal non-recombinant gamete, one type, non-recombinant gamete with pedicentric inversion, one, and recombinant gametes, and those are non-viable. Okay, but we will not find any kind of dicentric bridge in this case. Now, what are the significance of inversion? They do not change the overall amount of genetic material, okay, because there is no loss of gene or nothing. So they will not produce any kind of genetic imbalance because all the genes are there but in different position. Individuals with inversions are generally normal. Chimpanzees are normal, humans are normal, we are all normal. We have the inversions in our body. So, okay. And if there is no breaks in genes, if a break that disrupts the gene products, a mutation may be detectable in abnormal time. If there is some break in the inversion, then we can find out the mutation. What is mutation? Something different character than the normal. If the gene has an essential function, then the breakpoint acts as lethal mutation. Generally, in case of any kind of active gene, if that gene is not functioning properly, and if the body's repair system cannot repair that part or cannot prevent that thing, then generally it is lethal. And multi-deletion, multi-gene deletion, multigenic deletion in case of inversion or any kind of thing, it is lethal. And many inversion can be made homozygous. So inversion in both the chromosomes. Deletion to deletion. dicentric bridge. D. Pericentric or paracentric? Pericentric. I'll say paracentric. Because in case of this one is paracentric. Pericentric inversion, there will be no dicentric bridge. Okay, now the next part, the fourth one and the last one, is that translocation. What is translocation? Transfer of chromosomal segment between two non-homologous chromosomes. That is called translocation. There are three types. One is non-reciprocal translocation, reciprocal translocation, and Robertsonian translocation. So at first, think about the non-reciprocal translocation. We can denote the chromosomes like this also. This is another way to denote the chromosome, particularly for this kind of patterns, that is inversion, deletion, duplication. So the dot means the centromere, and those are the genes. So A, B, C, D, E, F, G, that is one chromosome with the centromere between A and B. M, N, O, P, Q, R, S, that is also another chromosome, and the centromere is between N and O. The EF part is translocated from this chromosome to that chromosome. Okay? So two different chromosomes, those are non-homologous chromosomes, but the part is missing from here and going to the another chromosome. This is called non-reciprocal translocation. In such case, the parental chromosome will be deleted. So we can say there is a deletion in the parental chromosome and there is translocated chromosome. This is called non-reciprocal translocation, that is no exchange. Only one part is going there. So this is a simple thing, that there is some break here and the break part is going to another one. If there are two things exchange between two chromosomes, that is called reciprocal translocation. Why did I put non-reciprocal? This should be reciprocal. Reciprocal translocation means exchange of chromosomal segment between two chromosomes. That should be reciprocal, not non-reciprocal. Now the main part is that, so there is chromosomal exchange. If the segment goes from one chromosome to another one, that is non-reciprocal. If both exchange their segments, that is called a reciprocal translocation. But this is very interesting. This is called a Robertsonian translocation. What is happening here? If there are two chromosomes and they are acrocentric chromosomes, acrocentric means, what is that? What is acrocentric? Eccentric means there is no centromere at all. And telocentric means the centromere is at the telomere region, that is the end point of the chromosome. What is acrocentric? Acrocentric, submetacentric, metacentric. What is acrocentric?
More at the top.
Acrocentric means just below the end point. So in such case, one part is very small, that is called, generally called satellite, that part. It is very small, and other arm is very long. Okay? This arm is very long, here is the centromere, and this part is very short. If the translocation is happening between these two, so that means in this part there are very less number of genes. The satellite part, there is very less number of genes. If the breakage occurs in this short part, suppose in the gray one and in the orange one, there is a break here, even in the short part also. Two long arms joined together with the centromere. And these two short parts, generally, they are failed to segregate. That is very small. There is no such gene there. So one chromosome is forming here, having all the genes from two chromosomes. This is due to translocation, but the thing is that one, then it is creating some kind of very big chromosome of 21. And in case of human, and that can cause the trisomy of chromosome 21. 3 chromosome 21. Okay, this kind of things. So 14 and 21, these two chromosomes are together. There is a Robertsonian translocation, break in the short arm and one break in the long arm. Break in the 21, the long arm, and in 14 in the short arm. They join together, and in such case, we can have the chromosome 21 in 3 doses. Because this is long arm means all the genes are there almost. So they can apply all the proteins. That protein is in 3 doses. When the chromosomes are now in 3 in number, that is called trisomy. Okay. Due to translocation, there is a trisomy of chromosome 21, and that can cause Down syndrome. Now, what is happening for translocated chromosomes? During meiosis, they can pair up homologous chromosomes, but they have some translocated parts. They can pair up with the matching one, and they can form this kind of cross during meiosis. So one spindle is attached here, one spindle is attached here, and here is one spindle, here is one spindle. So there are 2 poles here, and they will join like this because these are the homologous parts. And then they need to separate. Now, these 2 can separate to that way, these 2 can separate to that way, that is possible. These 2 can separate to that way, these 2 can separate to that way, because if the polar spindles are here, from one pole the spindles are here, and one pole the spindles are here, they can separate in that way. Or if one spindle is attached here and another spindle is here, they can separate in that way. Based on that one, we can have different kind of gamete formation. And there are 3 different names. If they can separate in that way, that is called alternate segregation. In case of alternate segregation, we can find this kind of gametes. If they can separate in just up-down, that is called adjacent one segregation. Then the gametes will be different. That is required for if we think about the genetic syndromes, then we need to think about whether it is adjacent one, whether it is alternate, or adjacent two. Adjacent two means that vertically, they are dividing vertically, like that. So these are the 3 processes for separating the translocated chromosome during meiosis. Now we have another thing which is called fragile site. Fragile site means there in our chromosome, there are some parts are there which is very, very fragile. And if there are some kind of gaps, they can break very easily. And in our X chromosome, we have this kind of fragile sites, and sometimes this can break. If this will break, then there is a chance of disorder. All the genetic diseases are disorders. So they can have a chance of disorder, and mostly the disorder is mental retardation or some kind of neurological problems. These are the main things for any kind of genetic disorders. Okay? And it is about 1 in 5,000 male births. So it is not very less. 1 in 5,000 means it's a very big number. If you consider the 8 billion population, right now probably we are reaching 8 billion right now. So that is quite a lot. This kind of syndrome is called fragile X syndrome. Okay. And we have more than 100 fragile sites identified in human beings. So there is a very good chance that having that fragile, this kind of syndrome. Fragile X is very common and it is from, for a long time we know that Fragile X syndrome. But in case of autosomes, they could be some kind of problematic also. But generally there is translocation and duplication of the genes. What is the outcome of a Robertsonian translocation? Two acentric chromosomes, one metacentric chromosome, one chromosome with two very short arms, one metacentric chromosome and one acrocentric chromosome, two metacentric chromosomes. Robertsonian translocation. See, see what B, B, okay, so I'm just giving this one. Next day we'll start this one. So we have structural aberrations. That part is done. Okay. Now we need to know about the numerical aberrations. Numerical aberrations are of two types. Either one chromosome is more or one chromosome is lost. The whole set is there. Now think about the whole set and one chromosome. We have 23 pairs of chromosomes. The whole set means 23 pairs of chromosomes. It could be 23 into 23, in 23 into 2, 23 into 3, something like that. Or only one chromosome. Suppose chromosome number 23, it should be two chromosomes, there are three chromosomes. If that is the case, only one chromosome is lost or excess. That is called aneuploidy. If the whole set is lost or whole set is excess, that is called the polyploidy, euploidy generally. If one set is lost, that is called monoploidy. If the whole set is more than 2, that is called polyploidy. Aneuploidy means nullisomy. Nullisomy means suppose chromosome number 14, the 2 chromosomes are deleted totally. It is not in the gamete. That is not in the zygote. In such case, that is called nullisomy. That means that chromosome is totally lost. 2n is the normal, that is we are diploid, and minus 2, that 2 chromosomes are missing. Monosomy means only one chromosome is missing. In case of Turner syndrome, we call XO. Only one X is there, another X chromosome is missing. But whole sets are there, 2n minus 1. So we have 46 chromosomes. We could have, in case of Turner syndrome, we will have 45 chromosomes. That is called monosomy. Trisomy in case of haploid. In case of haploid, there is n numbers of chromosomes, but one chromosome is double. That is called disomy. Only one chromosome is double. Trisomy means generally the Down syndrome, chromosome number 21 in 3 doses. That is called trisomy. Tetrasomy means suppose chromosome number 21 in double 4 doses. That is called tetrasomy. In case of human, trisomy is a common genetic disorder. Monosomy is also there, monosomy and trisomy. But in case of plants, we can have all these kind of things. It is very common in plants. What is polyploidy? Polyploidy means number is doubled. So we generally have 2 sets. If it is 3 sets or something like that, that is called a polyploidy. Triple. So 23 into 3. So 46 plus 23, that chromosome. That is called the polyploidy. Based on that one, diploidy, it is the normal one. So we have two sets of chromosomes. That is the normal one, that is called diploidy. Type means two. Triploidy means three sets of chromosomes are there. Tetraploidy, four sets, and such on. Pentaploidy, hexaploidy, heptaploidy, but it is very common in case of plants and in case of some peas. There are these ploidies, but in human, we generally don't find this kind of ploidies there. Now the question is there, we are diploid. If one set is totally missing, then the number should be n, from 2n to n, one set is totally missing. In such case, it is called monoploid. Okay, one set is missing. Now in case of haploid, when we are producing gametes, those are haploid. That is also n. And this is monoploid is also n. So what is the difference between haploid and monoploid? This is your question for the week. Okay, you need to find out. Next day I will ask this question first. What is the difference between monoploid and haploid? Both are n. And next day, actually, this is the main part. We need to work on non-disjunction. So if you go to the slides and just look at the non-disjunction part, then it will be easier for you to understand. Because based on non-disjunction, we need to develop the Klinefelter syndrome, that is, study the Klinefelter syndrome, Down syndrome, and all of it. Okay. Again, meiosis and non-disjunction. I'll give you the homework, post it in Canvas, and I'll let you know. Okay, another thing is that do you have any scanning software in your phone, like Cam Scanner? Cam Scanner is that is free software, free, that is app. You can download in phone. In such case, if your homework, you can do that one, you can just scan it and post it in Canvas. Okay? Because taking the picture, JPG is not very good. Actually, I cannot read that one. But in Cam Scanner, you can have the really scan there. And it is available in Google and iPhone everywhere. And it is free. So you can download it. In such case, just scan your pages and put it there. And in genetics notes, I have posted a lot of things. Okay? Because you didn't do anything, so I had to do that one. I told you to add something there each and every week. So today you learned about the structural evaluations, all those things, about some points there, and let them others do that. You can add 3, they will do 3. And they will add 3, you need to do that one. Do it in that way. Then you need not to study too much during your exam. Because the midterm and final will be 50-60 points, not like 20 points. Okay. So do with this one. Study your topics for the project. And I will, only for these 4, I need to change the group, otherwise the group is same. And Lynn, you need to find out any of these persons, any of these groups. There are 3 groups there, and there are 3 persons in each group. So you need to talk with them and add in one group. So one group will be 4, other group will be 3. That is fine. Okay. So just talk with them in which group you are in. Just let me know. Okay. And the topic is there. You need to work on that topic and do the studies, update the pages, update the PowerPoints. I will always check that one. So rather than discussing, and another thing is that if you have some kind of problem of getting those kind of things, every day after this class, we can have a short meeting about the project. That is the initial part we need to discuss. So just let me know, so we can have some part, all the group members and me will discuss and we'll talk about that one. But at first, go through the topic, to the Wikipedia, internet, whether it is, just go through the topic, have some knowledge about that one, basic knowledge. Then we will discuss about it. How to present, what are the informations you need to collect, what you need to show, all those things. Okay. And almost after each and every class, I can have a meeting with one group every day. So that is not a problem. Just 5-10 minutes. That is quite enough, I think so. But I need a solid project. that even you can present here when we have the student presentation. You can present here also. That kind of proof. Okay. Real scientific proof. Try it. Try it. Go for it. I'll always say go for it. Okay, so we are done.